Welcome, everybody. Please take a moment to type in the chat if you can hear me. It'd also be nice to hear where everybody is joining us from. It's great to see everybody. Please make sure that when you send your chat, it's to all panelists and attendees. All right, welcome everybody. This is our first Organization of Biological Field Stations live from the field event of the fall semester. My name is Kerry Wininger with Sonoma State University's Center for Environmental Inquiry. After a successful event last July, we have 3 more planned this fall and encourage you to take a look at the events lined up for October and November as well. Very quickly, some guidelines for the Zoom webinar. Remember, all participants are muted and your video cameras are off. During the presentation, please use the chat button to communicate with the panelists and attendees. Remember to choose all panelists and attendees so that everyone will see your comments. During the presentations and the live Q&A session with researchers, please submit your questions using the Q&A button, not the chat. And if you're here as part of a college class, please type the word student in front of your question so we can prioritize your questions. And it's important to make sure your full name is visible, either as your Zoom username or typed in after the word student, so that you receive credit for attending today. Also, welcome if you are watching us streaming live on Facebook. We will try to get to your questions too. Without further ado, I will turn this over to Beth Norman, Director of Science and Research at Lacawac Sanctuary Biological Field Station and Environmental Education Center.
Thank you all for joining live from the field. We welcome classes, faculty, and other participants from across the nation. Welcome, everyone. This is the second Live from the Field event. In this series, we take you on virtual field trips to research sites around the country and the world. As you will see today, we are visiting 3 biological field stations, places where people from all backgrounds come together to study the environment. Today, we are traveling to field stations in Ohio, Pennsylvania, and Kentucky to talk with researchers studying harmful algal blooms in freshwaters. Cyanobacteria are part of the phytoplankton or algae community in many freshwater ecosystems. Some cyanobacteria are capable of producing toxins that are harmful to humans, pets, and wildlife. Blooms of these cyanobacteria, often called harmful algal blooms or HABs, are occurring more frequently and are an emerging environmental and public health threat. Each researcher has prepared an 8-minute video outlining their HABs research. After the videos, they will all be available to answer your questions live. I'd like to encourage everyone to post your questions in the Q&A as we go along. Students, remember to type your full name in front of your question. The Stone Laboratory is located on Gibraltar Island in the western basin of Lake Erie. Lake Erie experiences frequent cyanobacteria blooms that can have major impacts on regional recreation and drinking water. Dr. Justin Chaffin has been a researcher at the Stone Lab for 8 years, specializing in the ecology and physiology of cyanobacteria. Dr. Chaffin's work allows him to enjoy lakes while solving important environmental issues. He joins methods he uses to detect cyanotoxins in Lake Erie.
My name is Justin Chaffin. I'm the research coordinator at Ohio State University Stone Lab. We are located on South Bass Island in Lake Erie. My job title is a research coordinator, and I specialize in harmful algal blooms in Lake Erie, and now we're doing some projects in inland lakes. The harmful algal blooms are a type of algae that can produce toxins, and my projects focus on what causes the blooms to grow and what causes them to produce toxins. And we're using that information to alert water treatment plants that pull in water that need to remove the algae and remove the toxins so they can produce safe drinking water. How we do this, we do a combination of field sampling. So we're out here on the research vessel today. We'll grab samples from the lake and process them and get data that way. We also do experiments where we'll get water from the lake that has algae, take it to the lab, incubate it with extra phosphorus or extra nitrogen at different temperatures and see how the harmful algal blooms respond in terms of growth and toxin production. There's two ways we get data from the lake. One is through deployed instruments. So you see a buoy behind us. That buoy has a water quality sensor that is constantly taking water temperature, water pH, turbidity, the amount of oxygen, and also the amount of algae and amount of harmful algal blooms. That is constantly recording data. The data gets beamed to the internet and anyone can download in real time. We also grab water samples, which we'll demonstrate here next, where we'll grab water sample, process it, and take to the lab and get more detailed data, such as how many, what type of algae are present, are there toxins present, what are the nutrients that are in the water, how much sediments are in the water. So we're going to demonstrate that now. One way we collect water from the lake is using a device called an integrated tube sampler. This will give us a sample from the surface down to 2 meters. And it gives a good representation of what's happening at the surface. We simply don't want to throw a bucket over the water, over the edge, and scoop up surface water because it might be a little biased. If it's calm, you might have a lot of algae floating at the surface. If it's rough, that algae might be mixed further down. So we want to get a nice slice of the entire water column. How this works, we're going to lower it into the lake, nice and slowly. Then I'm going to cap it, and this is analogous to like putting your a straw in a glass of water, putting your finger on the straw and pulling it out. The water remains in the straw. Water is going to remain in the sampler, slowly pours out. So now we're going to test the water sample for microsystems, which are the main toxins that the cyanobacteria produce. This project, or this device we're working on, is a new instrument that will allow us to get toxin data in 20 minutes. The normal method of analyzing for microsystems takes about 4 to 5 hours, whereas this will give us data in about 10 minutes. So the first step is to lyse the samples or to break open the cells. One of our projects that we're working on is testing a rapid microsystem test. The normal microsystem test takes 4 to 5 hours and has to be done in the lab. So we're working with this company based in Boulder, Colorado, that's developing a rapid test that'll get data within 20 minutes. So the first step of the method is to break open the cells, which we did. It takes 10 minutes. And then next, we are going to put 100 microliters of the sample onto a cartridge here. This cartridge has all the same chemicals that the standard lab test has. So the next step is to add the lice sample to the reagents, which you see Autumn here doing now, pipetting the water and the reagents together. And then once that's mixed, she will add it to the cartridge. So she's adding the sample to the cartridge, and then she will pop it into a reader, which then takes 10 minutes. All right, now the sample will take 10 minutes. A couple of the research projects that we're working on are funded by NOAA through their EcoHab program and their MIRHAB program. The MIRHAB program is the one that I talked about using the rapid microsystem tester. We're doing that to get a lot of data. So we need a lot of data in the lake to populate our models that are, which is the second project that we're working on, is funded by the HAB program, where we're taking all the data from the lake, putting it into a model, and trying to forecast harmful algal bloom toxins several days in advance. So water treatment plants, beach managers, general tourists have an early warning or an early idea of how many toxins will be in the lake at that given time. The test is finished. The Microsystem concentration was 0.8 micrograms per liter. That's pretty typical for this time in Lake Erie in the Western Basin in late August. We've seen much higher. The swimming advisory is 6 micrograms per liter. So the water here is still perfectly fine to swim in, perfect kind of fine to fish. This also, what I did mention earlier, this test also does a second toxin called cyanobacteria, which we do not see very often in Lake Erie or in the state of Ohio. But this instrument does have the potential to do, or it does do 2 toxins at once. Just for our purposes, we're only concerned about microsystems because we will never see, or hopefully we'll never see, cyanobacteria. So then what we would do, we would take that sample we collected, take to the lab, and do the gold standard ELISA test. So we can compare 0.8 from the N-bio unit to what we would get in the lab.
Now we travel east to the Pocono Mountains of northeastern Pennsylvania, where I work. This region has over 150 lakes, all of which are, of course, much smaller than Lake Erie, but are vital to the recreation and tourism-based economy. Lacawac Sanctuary's Pocono Lake Ecological Observatory Network, or PLEON, works with lake communities to monitor water quality and has partnered with researchers from Miami University to address HABs. Lauren Knows, a PhD student from Miami University, will be talking to us about her research investigating the occurrence of HABs in the Poconos. Her 4 years at Lacawac has allowed her to connect ecological theory with practical experience. Please bear with us with the background noise for some parts of this video.
Hello, my name is Laura Knows, and I am a 5th-year PhD student at Miami University in Oxford, Ohio. And I study under Craig Williamson with his Global Change Limnology Laboratory. So my research is focused on limnology, or the study of lakes. In particular, I focus on harmful algal blooms. I got into studying lakes because I grew up on a lake. It raised me. Lake Springfield in Springfield, Illinois is my home lake. And even though it wasn't on the guidance counselor's list in my high school, I knew I wanted to study science and become an ecologist. So I went to Miami. I did my undergrad at Middle Tennessee State University in biology and chemistry. And I did a couple of years getting my master's in public health so I could focus on the human aspect and the interconnection between how humans and the environment influence each other. I worked for a few years as a contractor to the US EPA, working on their health impact assessments. I decided I wanted to come back to school, get my PhD, and learn more about science.
There are many aspects to being a graduate student. The classic, of course, is taking graduate level courses. But also we get to perform research and lead our own research projects. I am leading a few research projects in particular related to harmful algal blooms. For one, I am managing and deploying the Arthur buoy, which is the aquatic resource tool for high frequency underwater research, also called Arthur. And it has a suite of sensors that drop into the lake and measure the changes in phytoplankton community assembly, particularly the total algae by chlorophyll fluorescence, as well as how much cyanobacteria or blue-green algae are in the lake. You can do a similar type of measurement by going out, as I do every month, and grab samples from different layers of the lake, and I measure how much algae are in those samples. So historically, there haven't been
a lot of monitoring for harmful algal blooms in the Pocono region. So there hasn't been much need to develop a centralized or a statewide monitoring approach until recently. We've been getting more emails and phone calls and just notices that observations of residents in the area or visitors to the area seeing algal blooms occurring later in the summer, longer in the year, and occurring more often. So this really led to my question, well, just how often are harmful algal blooms occurring in the Pocono region? What is the extent of harmful algal bloom occurrence here? And as well as what's the presence of toxins in the area, in those lakes? And I've partnered with the Pocono Lake Ecological Observatory Network, or PLEON for short, to go out and sample a suite of lakes in the region and try to identify the occurrence of harmful algal blooms in these lakes. In addition to that, we're looking at a suite of environmental conditions that could give us an idea for why harmful algal blooms are happening, such as the transparency of the water, the temperature of the water, the level of nutrients in the water, that could give us a better understanding of what is happening in these lakes specific that is favoring these potentially toxic algae. As with any project, you want to try and anticipate your challenges before you start, but you also tend to run into challenges while your project is going. For example, you need to know what funding you have available to complete your project, how many people, as well as the technical capacity or personnel expertise that you can tap into. As well as you want to design or figure out what's the best way to collect your data. For example, a lake, if you sample right near the shore, you could get a completely different picture than if you sampled in the center of the lake. As well, across spatial differences, there are temporal differences. For example, conditions in the lake may change very quickly from morning to the afternoon, as well as from day 1 to day 2, and month 1 to month 2. So one thing you have to consider is whether you're going to sample one lake several times to get a better idea of what occurs across a temporal space in that lake, or you could sample several lakes at one point to get a picture of what's happening in the region. And you have to do that balancing act between what information you really need versus what information you can get based off of the resources that you have. So with our Pocono Lake Ecological Observatory Network survey study, we had to anticipate these needs as well as the challenges we may face, for example, the timing and funding that we have. And we took a kind of an alternative approach where we used both techniques, where we sampled several different lakes at a single point in time, but we sampled in 2 different parts of the lake, such as the near shore and the center of the lake. So we could get a better picture of the variability within the lake as well as across the region.
So there are many different ways in which you can detect and monitor for harmful algal blooms. In our particular study, we use a variety, as many of the methods available as we could, so that we can detect the best way for a particular lake given its specific. So one of the ways you can detect for harmful algal blooms is a simple visual check. Is there a general discoloration of the water? Is there localized scum or surface collection forming? Is it general or is it localized? And then you can actually measure the changes in fluorescence or the pigments that are occurring in the water using a C6 or a fluorometer that measures the fluorescence of the signatures coming from the phytoplankton themselves. You can also use this very technique by analyzing satellite images, as well as grab a sample of the phytoplankton and take it into the lab. Into the lab, we looked at the presence of certain potentially toxic cyanobacteria, as well as the general population of phytoplankton in our sample, and we tested for toxins using ELISA or molecular methods.
All you need is some proper protective equipment like gloves, a well-labeled sample bottle, and this is just what we do in research projects. Grab, literally, a grab sample under the water, make sure it's fully submerged, no bubbles, and you can put it in a baggie, a cooler with some ice, and ship it right to the lab and get it analyzed. And we can look at not only what types of phytoplankton are present, what the relative abundance is, so how many of one particular group is more dominant, as well as other chemical analysis, such as are there toxins present, what levels, and nutrients and other types of information can come from just one little sample model. The main vehicle that most undergrads participate in research is they reach out to the graduate students in their program or in their university and ask them about the research they're conducting, or ask their professors about research they're interested in. And they can volunteer or get work study permits to work in those labs and get some experience that way.
Our last stop is in Kentucky at the Thomas More Biology Field Station on the banks of the Ohio River. The occurrence of HABs in the Ohio is a relatively recent development, but a very important one, as the river is a key commercial waterway and a critical source of drinking water. Dr. Chris Lorentz has been working at Thomas More University for 27 years, during which he has been able to involve many graduate students in fieldwork. He and undergraduate researcher Haley Oldfield will discuss the development of a HABs monitoring network for the Ohio River, how HABs are detected on the river, and the role field stations can play in responding to emerging environmental challenges like HABs.
Hello, thanks for joining us. I'm Chris Lorentz, Professor of Biological Sciences at Thomas More University in Northern Kentucky. We're a small, primarily undergraduate institution where I teach a variety of classes including Gen Bio, aquatic biology, and invertebrate zoology. I'm also director of our biology field station out here. For those less familiar with the field stations, they are typically associated with colleges and universities. There's plenty of private ones as well. And we take on all sorts of shapes and sizes. But generally speaking, we share similar facilities and similar missions of research, education, and outreach. Here's a look at our lab buildings, where we conduct research on the water quality of the river and propagate endangered and threatened aquatic species. We have a STEM outreach center for K-12 schools and the general public. And up on the hill, we have housing for student interns and other visitors. Field stations and marine labs are great places to learn content and develop research skills. But equally so, they offer unique opportunities for students to develop a deep sense of place about the environment, develop a strong sense of community with other students, faculty, and researchers, expand one's professional network, and explore career paths. Though we share similar missions, we tend to take on the flavor of our local environment. Being right on the Ohio River, our focus is on rivers and other freshwater ecosystems. Let's take a closer look at the Ohio. Here we are on the banks of the Ohio. You can see our STEM center and lab building in the background. And the river is about 1,000 miles long. It starts upstream near Pittsburgh, Pennsylvania, and flows generally southwest to Cairo, Illinois, where it spills into the Mississippi River. We're about halfway down the river near Cincinnati, Ohio. The river is a major transportation artery for commercial traffic, a source of drinking water for nearly 5 million people, and a source of recreation. Lots of boaters and fishers enjoy the river. It's also a rich ecosystem, supporting over 125 species of fish, about 60 species of mussels, and many other aquatic organisms. Much of our research revolves around monitoring the aquatic community to assess the ecological health of the river. Having dams on the river, it's a unique blend of allotic ecosystems, flowing water, and atlantic ecosystems, still water. It's really a hybrid between rivers and lakes and supports a rich plankton community, including blue-green algae or cyanobacteria. In 2015 and again in 2019, the Ohio River experienced significant harmful algal blooms, which was new to us. HABs are common throughout the U.S., but more often on lakes, ponds, and other slower moving bodies of water. Ours took us by surprise. The one in 2015 started near Huntington, West Virginia, moved upstream towards Pittsburgh, downstream past us here in the Cincinnati area, and down to Louisville, Kentucky. In total, it covered over 600 miles. Microsystem aeruginosa was the common blue-green species in the bloom. It dominated the samples and produced toxins that led to fish kills, disrupted the drinking water utilities, and limited contact recreation on the Ohio. Overall, it had major impacts, which prompted us to quickly form a harmful algal bloom detection network consisting of scientists from academia and governmental agencies. One of the outcomes of our HABs collaborative network is this early warning detection system, as you can see here. We have a solar panel that charges this battery in the box, which powers up the camera. And this camera takes a photo of the river every 20 minutes, sends it to our website where it's run through an application. The application was developed by a colleague of ours at Northern Kentucky University, Dr. Mike Waters, a math professor. And this application analyzes the pixels in the photo, can actually tell us the ratio of blue-green to brown algae in the river. So by taking photos of the river constantly throughout the day, we have a constant measurement of the amount of harmful algae in the river. Alongside the camera system, we conduct routine algal sampling to confirm what's in the water and also collect chemical data such as oxygen levels, temperature, nutrients to try and correlate what are the triggers that may lead to a bloom.
So my project right now focuses on harmful algal bloom detection, and a harmful algal bloom is a large proliferation of algae that can cause negative impacts to other organisms, people, and the broader ecosystem. So for example, this can look like fish kills, or even states closing off their fisheries, or even cities cutting off water supply to their residents because it contains some toxins. So last fall, there was a harmful algal bloom here in the Cincinnati, Northern Kentucky area that caused a halt in the recreational use of the river, and it also cost water treatment plants lots of money to filter their water. So here at the field station specifically, I am taking daily water samples, ratios of green algae and then yellow-brown algae to track trends and also predict harmful algal blooms at the same time. So agricultural runoff is full of fertilizers, pesticides, and herbicides that can end up in our waterways. Specifically, fertilizers constitute the majority of agricultural runoff, and they're full of nutrients such as nitrates and phosphates that can really spur the growth of algae. So when this happens, the algae will form a large mat over the water body, which will block sunlight from reaching the water column, and then that causes a depletion in dissolved oxygen. So when this process happens, it's called eutrophication, which constitutes it as a type of harmful algal bloom. And also depending on the strain of algae present in the water body, it can release toxins when the cell lyses. So that can just be another added negative impact from eutrophication and harmful algal blooms with agricultural runoff.
Our work on harmful algal blooms is relatively new to us. We've been collecting fish and water quality data since 1967, but when the blooms occurred, we quickly pivoted our focus to algae. Right now, we're trying to answer basic questions like what are the conditions that led to the blooms? What is the influence of climate change and the impact of biological factors such as invasive species? Though working with algae is relatively new to us, we were able to adapt and pivot pretty quickly, given our prior experience on the river and our understanding of the ecosystem. And this adaptation and flexibility are really hallmarks of field stations and marine labs. Being immersed in the environment, having long-term data sets and a deep understanding of their ecosystems allow us to adapt and be flexible and address some research questions as they arise. Whether it's harmful algal blooms, emerging viruses, or climate change, we can really shift and move in directions as the science dictates. Thanks again for joining us, and best wishes for your fall semester.
It is now time for the panel discussion and Q&A. Please post questions for the researcher panel using the Q&A button rather than the chat. Our college classes have a time constraint of 50 minutes, so we are initially going to give priority to their questions. After that, we will answer questions from other attendees up until the top of the hour. As a reminder, students, please type the word student before your question and make sure your full name is visible. The panelists are going to try to keep their answers to a minute or less so we have a chance to get to some of these great questions.
Thank you, researcher, for all of that wonderful information. To get us started, I'd like to hear from all of the panelists, briefly, on the same question. So let's start with Justin Chaffin, then Lorentz, then Haley Oldfield, and ending with Chris Lorentz. So remind us, what drew you to HABs research in the first place?
Hey, this is Justin Chaffin. What drew me to HABs was I always had an interest in the lakes. I grew up about 5 miles south of Lake Erie, and I attended Stone Laboratory as an undergrad, as I saw a flyer for it at my college, and then I got into grad school wanting to study lakes, but my advisor at the time had a grant to study harmful algal blooms in Lake Erie. I really enjoyed the project, and that's how I got into it, kind of by default. But I could have been I could have been a fish person or a zooplankton, but I ended up studying HABs.
Hey, hi everyone, my name is Lorentz, and I'm glad to be part of this panel discussion about harmful algal blooms. I actually started my first year working on zoonotic parasites, so parasites that can be passed from wildlife to people in lakes, and other types of waterborne diseases caused by other types of bacteria. But through a lot of community meetings and presenting on my research, I found out that most of the community was more interested in harmful algal blooms in this region, and seeing these HABs occurring more often, more frequently. We're really concerned and wanted help figuring out what to do about harmful algal blooms. So because of the community's interest and desire, I switched to harmful algal blooms, and I've never looked back. So it's been great.
Hi everyone, my name is Haley, and my research with algae began last year when I was a freshman with my undergrad. I was kind of thrown into working with phytoplankton, and then last summer I was lucky enough to work at the Thomas More Biofield Station looking at harmful algal blooms. And kind of the one thing that I like about algae is that it changes every day. I went in the lab and I never really knew what exactly I was going to be seeing, so it kept me on my toes, and it's interesting being able to see things that people don't see with their naked eye and that they don't know are in the water, and then be able to see them and identify them and kind of draw some conclusions from that.
Yeah, our research with algae really came to us. We've been working with fish and mussels for over 50 years, but with the 2015 bloom, the magnitude of it and the potential impacts of it really compelled us to shift and pivot our focus to algae. And fortunately, with our background of the river and also with our great colleagues in the area from governmental agencies and other academic institutions, we were able to quickly form this collaborative detection network team. And it's been great working with others on this common issue.
Well, thank you all for those answers. Let's start with a very foundational but important question. And I'm going to give this one to Lauren Nose. A student, Chantel Walker, asks, are all algal blooms harmful?
Oh, I love this question. So I do want to say that a harmful algal bloom, the name in itself is kind of a misnomer, because there can be algal blooms that are harmful but not toxic, and that they can have phytoplankton or the algae in such high dense populations that when they finally die off, microbes that degrade or decompose the algae then suck out all the oxygen, which can lead to en masse die-offs of fish and other aquatic animals because of the lack of oxygen, not necessarily from toxins, and also from clogging of gills. But typically and most commonly when we refer to harmful algal blooms, we're talking about those that are toxic. So it's important to note that just the discrepancies, little nuances between the terminology.
Thank you for that. So next up, I'm going to direct this question at Justin Chaffin. Were these the algae that killed about 300 elephants in Africa? Are they the same type?
Most likely. I probably read the same news article that the student read. Likely cyanobacteria, but the article did mention neurotoxins were present in the water. The neurotoxins are the toxins that are associated with dog kills in the United States. So it's probably that those neurotoxins. But in order to confirm, you would need to do autopsies on the elephants to confirm that it was toxins that killed them, not something else, but likely it was cyanobacteria and a neurotoxin.
Yeah, I just wanted to add real quick to Justin's comment is that there are several different types of species of cyanobacteria, and there are some cyanobacteria that don't produce toxins and some that do, and some that can switch between toxin production stages and non-toxin production stages. So it can be really tricky in deciding whether it's a toxic bloom, and hard to decide which species it is because the same toxin can be produced by different species as well. So it's really best to look to see what type of cyanobacteria that it was that was in the water, and you can do some further testing molecular-wise to decide whether it had the genes to produce the toxin, and then of course testing for the toxin itself.
And you know, that leads right into a question here that I'm going to direct at Chris Lorentz. It says, here at Clear Lake in Lake County, California, we've spent a lot of time on public education, focusing on the difference between cyanobacteria and true algae. I see you're calling cyanobacteria a harmful algal bloom. Is distinguishing cyanobacteria from true algae an issue for you?
Yeah, that's a really great question, and it's a really philosophical one. So by definition, these algae we're talking about are cyanobacteria. So that means they belong to this domain prokaryotes. They're primitive cells, lack a nucleus, and they're true bacteria. We've always called them blue-green algae because they look and act like algae. So they live in fresh water, they're unicellular or colonial, and they're photosynthetic. They're actually the first photosynthetic organisms before typical algae. So by definition, they're cyanobacteria, and there are many in the field who prefer to use harmful cyanos as the true descriptive term of these guys to A, be more accurate in terms of their taxonomy, but B, also distinguish them from green algae and other algae that are non-toxic and a typical part of the community. So that's a really important distinction. We tend to call them harmful algae, but in reality, they're cyanobacteria.
So this next question I'm going to address to Haley Oldfield because it is from an undergraduate perspective. So a student is wondering, how can I, as an undergraduate, become a part of this research? They've always been interested in working in the field as an environmental science major. So maybe, Haley, you can share your experience, and if any of the other panelists want to add in after, that would be great.
Sounds good. So my research kind of began my freshman year. My school in particular, they have like certain research courses you can take as a part of your like central schedule. So I was able to sign up for them like any other class. And then last summer, I was just like kind of looking around online to see what kind of things were out there that I could apply for. And that's when I found Thomas More's field station internship program. I'm originally from Kentucky, so I live like kind of close to the field station and close to Thomas More. So it ended up working perfectly. It's just a matter of kind of just doing some looking around online, seeing what kind of things are out there, and kind of looking around to places that you wouldn't expect there to be research. Like, for example, like I go to school in South Carolina, but I did an internship in Kentucky. So it's kind of like there's something, definitely there's things everywhere around the country, no matter where you're at.
Yeah, I would concur. Haley was a great intern for us. And if you look early, look in the fall semester, internships are typically posted for undergraduates, research experience for undergraduates, and a whole host of others. There's plenty of websites online, but also talk to your professors. They will give you some good advice on where to look and how to apply. But typically the applications are due in early spring semester. So use winter break, apply for plenty of internships that are of interest to you. And they're great experiences for learning content, skills, techniques, but also developing a network for career development, developing confidence in the field of STEM, and just networking with peers like yourself. So I highly recommend summer internships in a variety of fields, and there's plenty from which to choose.
It's wonderful to hear how many opportunities there are at all levels here to work on this type of research. My next question, I'm going to direct at Lauren. And this student asks, what impacts might climate change have on HABs and the detection network?
So this is a really good question, and that a lot of people are looking at it right now, particularly my lab as well. But for the most part, you know, with climate change, we know that it's not just one thing happening, but a suite of things happening. For example, in some regions, they're seeing more rainfall events, such as in the Northeast, where the Poconos are located. So they're getting a lot more nutrient runoffs into the lake, a lot more of the other organic matter coming into the lake. And that's changing not only the nutrient regimes, but also the color of the water, the transparency of the water, and that can potentially change the dynamics or who's dominant in the lake in regards to phytoplankton. One of the most common things that have been studied is the effects of temperature and the rising increase in temperature on phytoplankton. And yes, because cyanobacteria have that higher growth optimum, or a higher temperature that they survive better at than their competitors, the other types of algae in the lake, that they can proliferate and even do very well when the temperature of the water gets, you know, at or above 25 degrees Celsius, or about 77 degrees Fahrenheit, whereas other algae tend to die off.
Yeah, and here in the Ohio River, we think climate change is playing a role in our harmful algal blooms. So we don't typically get them. We had a big one in 15, another big one in 19. And climate change effects in our area were warmer and wetter. And that flooding in the springtime brings in new nutrients, causes turbidity, and it causes factors that are conducive to cyanobacteria growth. So we're looking at climate change, as Lauren said, it's a complex range of factors, but we certainly think that plays a role.
Okay, the next question is for Justin Chaffin. How does the water purification process differ for water with HABs? So let's consider that question drinking water.
Right. There are many methods to treat the water for drinking. Ideally, what you do is remove the cell. If you can remove the intact cells or the colonies, you'll remove most of the toxins, because when you get high biomasses, most of the toxins are inside the cell. So if you can remove the cell through meshes, through chemicals that stick to the colonies and cause them to sink, if you can do that, you remove most of the toxins. What is harder to treat is the extracellular toxins. An example, what happened in Toledo, Ohio in 2014, there was a bloom out in the lake. The bloom got attacked by viruses that are unique to cyanobacteria. The cells in the lake bust open and the microsystems were released. Those dissolved toxins pass through all those pre-filters, and then those become a lot harder to remove. So when you have extracellular toxins, you need to use, there's a couple options, but the most common way is powdered activated carbon. It's kind of like charcoal that's ground up in really fine particles, and microsystems that are dissolved will actually bind to that charcoal, and then you can remove that charcoal. Another way is through ozone treatment, other oxidative methods, but ideally, if you remove the cell intact, you can remove the toxins. We're also doing research on bacteria. Microsystems get degraded naturally by bacteria. So if you know the bacteria or you know their enzymes that you're using, you could potentially add them into the water treatment process to further remove toxins.
Okay, for students that have to leave now, thank you for joining us. We will be emailing follow-up information. For other attendees, the panel will stay on through the top of the hour and continue to answer your questions.
Our next question is fascinating, and there's actually a couple people that have wondered something similar, so I'm going to combine these. I'm going to start with Chris Lorentz. The first question is, how about the challenges dealing with doing field research during COVID-19? And on top of that, also thinking about how COVID-19 might actually help reduce harmful algal blooms. Did quarantine prevent people from releasing excess nutrients into the water?
Yeah, two great questions. The first challenge with COVID-19 was a big one. And I must say that I'm grateful for our university administration to show faith in us in terms of our ability to implement some safety protocols and actually have an internship program this summer. We were really grateful to do that. So we took extreme measurements in terms of assuring the safety of our students and adding additional cleaning protocols and wearing masks and gloves on occasion as well. So we were limited for sure. We modified our protocols for sure, but we were able to get what we needed to get done, and we are grateful for doing that. As far as some effects of the pandemic on this topic of harmful algal blooms, you know, the biggest source of nutrients into our waterways is agricultural runoff. And we really didn't see the limitations of that during the pandemic. So we still had the same sort of agricultural activity in our region and around the country. So we did see some changes, of course, with the atmosphere and CO2 and things like that, but those factors that directly address or impact harmful algal blooms were less influenced or less diminished under the pandemic.
Yeah, I just wanted to say a little bit different of a story here in the Poconos, because the Poconos, if you haven't heard of them before, they're typically a vacation resort site for a lot of New Englanders, especially people from New York, New Jersey. So when the pandemic came through and a lot of people were either put on remote work or remote learning or not to come in to work at all, they fled to the Pocono region. And so it's going to be very interesting to look at the nutrient content to see if it's changed significantly from normal summer vacation time, because people were staying longer than anticipated, and more people were coming to the vacation sites and trying to get away from the city than previous years.
Actually, I'm going to follow up on that question to Haley Oldfield. Can you explain a little bit in more detail the connection between nutrients and harmful algae and algae blooms in general?
Yeah, so when algae want to reproduce, the kind of things they draw on from the water are nutrients predominantly. So with agricultural runoff and fertilizers specifically, fertilizers are full of those phosphates and those nitrates that once they get into the water system, that's what the algae will kind of use as fuel to begin to reproduce, and their colonies will grow significantly from that. So like it's like a positive impact. When fertilizers enter the water, they increase the algae.
Great. So somebody actually has an interesting question that is a little bit outside of this event, but I think would be nice to hear about. So I'm going to go ask Justin Chaffin, can you compare, contrast HABs in freshwater systems versus marine systems?
Yeah, that's a good question. I lately been getting more interested in marine HABs, you know, just for pure interest. There's a couple fundamental differences. In freshwater, harmful algal blooms are cyanobacteria, whereas in marine systems there can be the red tides, which are dinoflagellate. There can be brown tides, which are a type of diatom. There can be other types of harmful algal blooms in marine systems. So it's just not one type of group of organisms. The second difference, or another difference, would be their impacts on the ecosystem. In freshwater, you know, our main concern is protecting drinking water, whereas in marine systems, they're concerned about the food chain and people eventually eating the shellfish or eating the fish that are contaminated with the toxins. So, you know, that's a different route. Freshwater, we're more concerned about drinking water, whereas marine systems they're more concerned about the food web exposure, because right now people don't drink seawater. And then I would also say another difference, which might be a little obscure, but is the amount of funding that is available for marine versus freshwater. The marine systems get a lot of funding, whereas the freshwater systems get a lot less. Even though we're starting to get more funding due to the impacts in drinking water, but historically, harmful algal blooms have been associated with marine systems, and what they see in freshwater has been, kind of shied away. But until recently, there's more funding coming towards the freshwaters because we drink freshwater and we all need freshwater to survive.
That's a perfect segue because a student asked earlier on, how do research projects on this topic get funded? And I guess I'll go ahead and start with Chris Lorentz.
Yeah, so there's a lot of funding, or I should say relatively a lot of funding at the federal level. NOAA, as Justin pointed out, is a big funder of Lake Erie research. The NSF, National Science Foundation, is another funder of research. But then also locally, there's state funding and county funding because of the impacts, as Jason alluded to, on drinking water. But another aspect is on contact recreation. And so the funding comes at the federal, state, and local level, primarily at different governmental agencies, but then private foundations will be supportive of our research, particularly as it helps to develop undergraduate research.
All right, I think we have time for a couple more questions. This one is for Lauren Nose. I'm going to combine a couple questions here. Does your research focus on a particular place in the water column? So you demonstrated a grab sample from the surface. Are you focused on the surface? And a follow-up question is, just because one area of a lake has a harmful algal bloom that has shown to be toxic, does that mean the entire lake is toxic or vice versa?
Yeah, so I love these questions, and they are at the heart of why we did the Pocono Lake survey last year. One is obviously you can sample at every depth, at every location. Over a period of time, you have to balance your resources and the time you have available and what you need. With the Pocono Lake survey, we took grab samples from near the surface at different locations, so at the center and near shore of the lake, because we were looking mostly for harmful algal blooms at near the surface, because the focus is mostly not really for drinking water, but for recreation. And so people are typically swimming in the top meter of the lake. Other projects that I've been working on, and particularly the buoy-based project, which is related to Arthur, the buoy sensor system, takes a profile of the lake 4 times a day, all the way from the surface to the deep waters. And some of the most interesting things I've seen is what's happening at 13 meters deep. You wouldn't expect to see algae, but they're down there and they're grabbing that phosphorus and they're enjoying life. And what's really neat part of research or part of this field is looking at that dynamic between day and night and the shifts and how some of the phytoplankton are moving throughout the water column on just a daily basis. So that's a really fun research project that we're working on. So, and I'm trying to remember the second part of the question, Beth. What was that?
It was, so if one part of the lake has a harmful algal bloom or a toxic bloom, is the entire lake then quote unquote unsafe? How do you judge that?
Oh yes, so this really comes back to the size of your lake. So, and also the shape of your lake and how it's oriented. For example, and how deep the lake is in particular. There's a lot of factors that go into play here, but a lot of times what happens is when the algae aggregate or the cyanobacteria aggregate together and form those scum on the surface, they can be blown or concentrated in a specific region of the lake by the wind. Also, you can have as you saw in the video that we took, a very generalized a bloom occurring across the entire lake. Even though we were just looking in just one cove, the entire lake was that green. So it can change very rapidly where the bloom is and how concentrated it is and what it looks like. And one thing to note is that the toxins can be just as variable across space and time. So one thing to note is most of the toxins actually are biodegradable in water, so they will go away with time, but some of them take longer than others. So you may see a spike in one area, and either current or wind mixing, the toxin concentration could change, or very quickly. So.
Okay, so we have one more question that we're going to get to. We have a lot of questions that we did not, but we will be sending some follow-up answers here. But this final question is for Justin Chaffin. You study the lasting effects of these blooms after they've subsided. The materials they release while living and after death obviously don't just fade away. Is there a noticeable impact on the environment around and beyond where the bloom was last congealed?
That's a really interesting question, and a lot of it we don't quite know yet. In the short term, we know in the lake that microsystems generally don't hang around too long if there's beneficial bacteria in the lake that would break them down. We done studies last year to determine what is the rate that bacteria break down microsystems, and it's really fast. However, if the bloom hangs out into the colder temperatures or into November, December when it's cool, that bacterial activity will be a lot slower, and those toxins can persist much longer. But when you start, working your way up the trophic levels, what are the impacts of cyanobacteria on fish? A lot of these questions we don't quite understand yet or don't know. It's, studying lakes there's lots of factors that go into, the success of a lake, if you call it that. But cyanobacteria are just one issue. So there's lots of factors to untangle, and the impact of blooms on the following year or several years down, we just don't quite know yet.
Well, thank you to our speakers, Justin Chaffin, Lauren Nose, Chris Lorentz, and Haley Oldfield. Thank you to the Organization of Biological Field Stations, to the National Science Foundation for funding this project, and to all those behind the scenes who made this possible. We will be emailing all of you in the next day or two with follow-up answers to any questions we didn't address today, and there are several of them, a link to a recording of this event, a link to provide feedback and ideas for future events, what you enjoyed, other event themes you might be interested in seeing in the future, and suggestions for how to improve. Up next is climate change at the Land-Sea Interface on October 13th, then nutrient cycling in grasslands on November 9th. Please share these two upcoming events as well as future events with fellow students, friends, and colleagues. Thank you and goodbye from the field. Panelists, goodbye.
Thank you.
Thank you for joining us.
Thank you.
Bye.
Yay! Oh, Kerry, can't hear you.
We still do have a few attendees in the meeting. So this isn't a completely private session right now. So I was thinking we would want to re-log in and do a practice session. But if that's the case, then I would need to invite Kari because she isn't a panelist. So it looks like at least a few of us are available. So why don't we go ahead and do that?
Are you going to send an invitation then, or do we log back in?
You can, I'm going to just end the webinar and then just log back in the same way we did at the beginning. And I'm going to do that first and then send Kari so that we can all be in the webinar while I'm doing that. Okay.
Did you see the message from Margot?
Yes, and Margot is a panelist, so she'll be able to join us.
All right, I'll see you in a minute.
All right, so I'm going to end the webinar.